All right. When we look at this, they're giving you an analogy here of a hill. And you'll see they say figure one, you're looking at 650 meters up. That's the top of this hill, where this one's 1,400 meters up. That's the top of the larger hill. It says if you're going hiking the weekend, the option of hiking a mountain of 1,400 or 650, but you know that you have limited time because of other plans. Which mountain would you choose to hike? A, this or B? And the idea here is the smaller hill will be easier for you to get up and over, and therefore faster. You can go at a faster pace. So your choice there would hopefully be was A. Now, if you're hiking at the same pace, which of these mountains would more require more energy to hike up, A or B? And again, more energy, it would take A. Now, information in B, if you're rolling a ball up over the hill, and then when you're rolling, it says a picture above shows someone trying to push a boulder up a hill. In order for the boulder to reach its final destination down here at point B, it must be pushed to the top of the hill first, because once it reaches the top of the hill, it will roll down all the way by itself. A familiar scenario exists in chemistry. Before a chemical reaction will take place, energy must be put into the system. The energy needed a chemical reaction to move from A to B is known as the activation energy. So we've mentioned this several times over the last week, and it's important that you understand that the energy from where the reactants start here is, and and to the top of the first hill here, this energy is known as the activation energy. The energy required to go to the top of that hill. Now, when that ball starts rolling down, that the energy put in will come back out. So this is energy going into the system. Required into the system, where the on the other side is the energy coming out of the system. All right, that's the energy of activation, EA. Now, when you look at this diagram, it's a diagram we drew in class several times, and we looked at it and talked about it. If we look at this, is the energy, energy increasing in the system, and this is going, you can think of it as this here is where the reactants start, A and B, or if you're putting two together or mixing them up, or if it's taking one thing and breaking up into two, whatever the products might be. In this case, you're exchanging elements in order to make new molecules. What you end up, well, in case let me give you an example. If you took NaOH and you combined it with HCl, here you have four different atoms. This would be Na, and that's sodium. This is oxygen, this is hydrogen, and this is chlorine. This would be your A, this would be your B, right? So A plus B is going to yield, after the reaction, you're going to yield a NaCl, a salt, table salt, sodium chloride. So that would be B. I'm sorry, that's not B. That would be C. And then you're adding to that, what would happen. Remember our rule, we have to have in the end what we started with. You have the Na, you have the Cl, you need the OH and the H. So, of course, or at least I hope it's obvious to you, you'd have water. And that would be D. So this is a type of reaction that you might see here. NaOH, A plus B in the reactants, A plus B, yields at the end C plus D. So you start with reactants, you end with products. And by the way, this is a neutralization reaction, just like we did earlier on with phenolphthalein in the osmosis diffusion lab. You have A, B yielding C and D. NaOH plus hydrochloric acid, they're neutralizing each other. In the end, you get salt and water. All right. So with most chemical reactions in your body, as we said before, what you don't want is a reaction that's going to happen on its own. Every time there's a reactant, it produces a product. Now, does that happen? Yes, but do we want it to happen all the time? No. You want to have some kind of control. And the control is kind of embedded in the biological system, and that control is the enzyme. Without the enzyme, it takes this amount of energy to make it happen. That energy is not present. That amount of kinetic energy is not present within a cell. If you had that much energy in the cell would just break apart. It would boil. So, just as an example. So that is why this amount of energy is just prohibitive. It's not going to work inside a living cell. But when the enzyme is present, the enzyme is able to lower that activation energy, so the hill is smaller, so the reaction goes much faster. And as you saw in our latest lab setup that we went over on Friday in the last video, these enzymes can catalyze hundreds of reactions, thousands of reactions per minute. So where you go from no reaction at all to thousands per minute, because of the idea of creating a space that does not require as much kinetic energy to occur for the chemical reaction to occur. So, the classic example is if you had these two particles, A and B, and they had to come together to make C. A plus B equals C. And a good example of that would be hydrogen gas plus oxygen gas. We did this with our battery. When we did electrolysis, we broke apart hydrogen and oxygen. We put it back together, and what we made was water. It's not balanced. If we were going to balance it, we would put a 2 in front of a 2 as a coefficient in front of the H2 and the H2O. So when we look at this, if we have these two coming together, that's two hydrogens, and over here you have 2 oxygens. Now, how often is there flying around in here? If they have to hit in just a very specific way, if they just bounce into each other at this kinetic energy that's in this container, if they hit each other in the container, nothing happens. They're not hitting themselves with enough force to get them, or at the right angle, in order to get this reaction to occur. So what we did in the lab when we made water from hydrogen gas and air and oxygen in the air is we used a small flame. We added kinetic energy, which gave this reaction enough energy to get over that activation hill. Now that would not be very useful, right? That would not be very useful in the body, because when hydrogen and oxygen coming together to form water requires that much energy. A flame, you would catch on fire in order to get it to occur. But we could get an enzyme that had a place for both the oxygen and the hydrogen gas to come together. And when that oxygen molecule found its way, or the hydrogen molecule was found the enzyme and the enzyme caught it like a baseball cat in a catcher's mitt, and the oxygen's caught here, then simply by them bonding, the enzyme changes shape. It has a conformational change, and which allows the two to come together. So it literally brings them together. In such a way, and I'm just making this up as I go here, puts them together in such a way that one of the oxygens is able to take the two hydrogens and fly off with it. The environment's such that the amount of energy required to get these two to have a chemical reaction occur is a lot easier to occur. I mean, it's a lot lower. And because it's a lot lower, it happens. And what you're left with is the enzyme back to its original shape, waiting for another set of molecules, another set of hydrogen and oxygen to come in so that it can catalyze the reaction between hydrogen and oxygen to form water. And in the end, what you end up with for every two sets, right? Because if you look at the chemical reaction, you need two hydrogens and to make two waters, because when you take one oxygen, it goes off with two hydrogens, you're still left over with another oxygen. So then you need another pair of hydrogens that come in and make two of these. So what we end up with is two waters when we started with one oxygen molecule and one hydrogen molecule. They come together in a way to make two hydrogen molecules and one oxygen molecule. Two hydrogen molecules. There's one hydrogen molecule, there's another. Each hydrogen molecule has two, look at the subscript, two atoms. So two hydrogen molecules for every one oxygen molecule. And when when the enzyme brings them together in such an angle, given the amount of kinetic energy in the system, boom, the chemical reaction occurs, and you end up with two molecules of water. One oxygen goes here, one oxygen goes here. Two of these go with this oxygen, and the other two go with the other oxygen. And so that is how a chemical reaction would be catalyzed. So when we look at lowering the activation energy, it's about this fitting into these specific shapes, right? The shapes end up being a very important component of what's going on here. This hydrogen molecule has to fit in this activation site. This oxygen molecule has to fit in this activation site. So it's very important that the shape, right, ends up that the shape ends up fitting the substrate. In this case, this enzyme catalyzes a reaction between these two, so it has an active site for the hydrogen and active site for the oxygen. All right, and remember this green, the enzyme is a protein, so it's folds back and forth. All right, does it speed up or slow down the reaction when you compare what happens when you're using an enzyme versus when you're not using an enzyme? Well, it speeds up the reaction because it lowers the Hill, which is why they gave you an example. The example they did on the previous page, and it speeds up, right? Does it speed up or slow down the reaction? It speeds it up. And I just explained to you how it does it, but chemical reaction lowers the amount of energy required to bring these two together, and the way it does it, again, is by bringing them together in a specific angle that it doesn't need as much energy for the chemical reaction to occur. So that's this answer. How does the enzyme impact activation energy? It lowers the activation energy. And if you're wondering how I know that, it shows it right here. It goes, the activation energy was here at number 1, but now it's at 2, right? So the height of that hill is much lower. So it's, again, reading the chart, reading the graph, and seeing what the impact of that graph is. Based on your answers to questions A and B, what do you think the term catalyze means? And we've said it numerous times in class, but I'll say it again. To catalyze a reaction, let me just say it the way it's stated here. Catalyzed means the chemical reaction chemical reactions activation energy is lowered, so it goes faster. Another key component here is, please realize what we said earlier when we went over the pre-lab, when we went over the lab together, and I posted the notes. The enzyme doesn't change. The enzyme just captures the molecules, brings them together in a specific angle, so that the amount of energy required for the bonds, the electrons to be exchanged and broken and reformed is a lot less. And then once the products are formed, the enzyme goes back to its original shape, ready to pick up another pair of substrates. And remember, the reactants in this case are called substrates. Can you think of any other reactants or any other things that are examples of a catalyst that are not enzymes, right? So when we look at the example used in class and in your textbook, it's catalytic converter is the most common example of a catalyst used in everyday life, and that's when we burn gasoline, we don't always get carbon dioxide. What we end up getting a lot of times is carbon monoxide, which is poisonous, right? We also get some nitrous oxide or NOxes, which nitrogen with NO2 and NO3, which can be also very damaging, and we can also get other forms of hydrocarbons like methane, CH4, or ethane, or other kinds of gases that would do us damage. So in a catalytic converter, what we do is we take those exhausts from the burning of gasoline, we run it through this honeycomb that has platinum and palladium coating it. And the platinum and the palladium do very much what an enzyme does, is it allows for these gases that we don't want to get arranged and positioned in such a way as to allow the oxygen in the air to mix with them, or in the case of the NOx's, to break up, and again in these two examples, for the oxygens to hit them in such an angle as to produce gases that are not as harmful. For instance, nitrogen gas and oxygen gas, or carbon dioxide, or carbon dioxide in water, for instance. So this catalytic converter actually has really been a great invention. It's allowed us to use this catalyst, platinum and palladium, which are metals, in order to quickly change these gases that could take 10 years in the atmosphere to turn into something else, to turn them into gases that can easily be used. All right, so that's the example that's in the book, and that's the example we had mentioned in class. So when we look here, what do you know about keys? What are the characteristics distinguishing one key from another? So shape, right? The shape of the key, this shape will open one lock but not another, etc. Where are some places you use keys and locks? Can a key from one place open a lock from a different key? They can. Most of the time, no, right? But they can. You can have even something called a skeleton key, which opens up a bunch of different keys. There's, for instance, my key that I have for our classroom opens up my classroom, but there's a universal key that Ms. Perez has that can open up all the classrooms. So there are some keys, some enzymes can open up or match up as a substrate to a family of substrates just because of the shape that the active site takes on. So, for instance, if a substrate has this triangular shape, just as an example, it'll fit, and the enzyme will do its job, even if it's slightly the substrate slightly askew, even though the substrate might have slightly different shapes on the other end. The active sites match up. As you can see here, this would all match up in the enzyme because they're all family of molecules that fit into this enzyme, and this enzyme works with it. But most of the time, the substrate's very specific to the substrate. The enzyme's very specific to a substrate. But it does happen, and in fact, a lot of poisons work that way. Acetylcholine, which is an enzyme that fits, or as a substrate neurotransmitter that fits into a receptor in your cells, very specifically. The acetylcholine is a neurotransmitter that only fits in that receptor, one of the receptors that we looked at earlier in the year. That there is a chemical called a nerve gas that could take the place of that acetylcholine and actually bond to it much more strongly than acetylcholine does. And so it doesn't break down and ends up that it's a very deadly gas because of that, because it can actually match up and take the place of a molecule, of a substrate, matching the active site of that protein and that receptor. So there are examples of things that are very specific, where substrates or signal molecules are very specific to an enzyme or a receptor, and that some other mimic or some kind of molecule that has similar shapes that fit into that active site can cause a lot of trouble. But in essence, the very basic answer here is that the keys are specific. To a lock. If they weren't, they would not be very useful. If you had to say specific or general, you'd have to say specific. Again, there are exceptions, but in general, that has to be the most reasonable choice. Now, here again are the enzymes and the substrates. So, as you can see, here is a substrate. Has two very distinctive shape, this kind of rounded edge, and here's a triangle. Fits perfectly in here. And when it binds, the enzyme substrate, they bind and form through hydrogen bonding, and they fit very precisely. Then the enzyme starts to change shape. It changes the characteristics of the active site, which lower these substrates and this enzyme has their own energy because there's kinetic energy in the system. That energy then becomes enough for these two to separate and form new products, right? So maybe it bonds to water and then it makes hydrogen and oxygen. Maybe it bonds to some other molecule and splits them up. But in any case, you start with a reactant that's made of specific components, and then it binds, and what you get is some products, right? So you start with the reactant and with products. Notice the enzyme started this way and ends this way. The enzyme itself is not used up, but look, the substrate's used up. You have less green, right? Here you start with a green molecule that has two parts to it. Then you end up with an orange and a blue that are smaller, that don't have this shape. They have some other shape. So you're going to use up your substrate as the reaction goes on. You're going to make more and more product as the reaction goes on, but the enzyme does not change. Look in figure 5 above. How does the enzyme similar to lock and key? This fits into this space. So if this is the key, this is the lock. Or this is the lock and this is the key. This is the lock and this is the key. I don't know. It doesn't matter. The lock and key analogy is that the two shapes fit perfectly, and they're very specific. So the similarity is the specificity of the two things. In this case, substrate and enzyme. What terms they use in the picture of the enzyme that could be used in the following analogy? A lock is to key as what is to what. If a lock is to key the way an enzyme is to substrate. If the substrate is in the first image on the left of the series is a disaccharide such as sucrose. So if this is sucrose, which would be made of glucose and fructose, right? So this is one, it's a disaccharide, so this is one monosaccharide, this is another monosaccharide. The enzyme is doing to this disaccharide, it's splitting them, right? It's hydrolyzing them. Remember, that's how in biological systems you add water to break things up. So hydrolyzing or splitting the two sugars into two separate sugars. The disaccharide turns into two monosaccharides. The picture above is showing what is known in biochemistry as the lock and key theory. Explain why you think this theory is used to describe enzymes. Because the two fit into each other. So the reason is because the enzyme and substrate fit into each other as specifically as a lock and a key. All right, what are some examples of biology, chemistry class, or from other areas of your life where a specific shape of something is important for it to work correctly? Oh, there's a ton of things where, we've talked about, and I think a few of you actually brought this up as an example you wanted to use. And I used it just a minute ago, which is signal molecule and receptor. Also very specific. And we've used insulin as a signal and the receptor on the cell surface. Very specific. There's the example of neurotransmitters that I used earlier, where you have this neuron. It's a cell that that transmits a signal to another neuron in order to let you think and move. And of course it has a nucleus, a nucleolus, a endoplasmic reticulum, a Golgi body, ribosomes, vacuoles, all the things that we've learned, all the organelles that we've learned about. It's very specialized, a very specialized cell in your brain and in your spinal cord and in your muscles. And these neurons, what they do is they send a signal from one cell to the next across what's called a gap, right? Where the signal has a specific shape, and the receptor on the other end is connected to a channel protein. This is called a ligand. A ligand in this case, because it's a receptor and a signal. And in this case, the ligand fits into a spot in the receptor. And we talked about this, that sometimes the signals can open channels. The channels can be opened up because of a signal binding to the receptor. Or in the case of insulin, the signal can send a signal into the cell. So then what happens is that this signal binds to the receptor, which then opens up a channel and allows ions into the cell across the membrane, because ions can't move across the membranes on their own. And at the very next, because of that bonding, the channel opens up as long as the ligand's bound to it. The protein opens up a channel, and that channel allows calcium ion to flow through the channel, which continues to signal down the neuron. So these neurotransmitters are held in vesicles inside the previous cell and released as soon as you want to have a thought. And that is very specific and very similar to what's going on with enzymes and substrates. In this case, they're called ligands, but with the enzymes, they're called substrates, right? This is called a ligand. This is a neurotransmitter, where this is a disaccharide. But they still have that lock and key concept where the two shapes have to match perfectly in order for the function to work. In this case, what this enzyme is doing is breaking up the disaccharide into monosaccharides. In this example, the neuron signal, the thought, the feeling, the muscle twitch is being conducted down a pathway. So consider that the idea of lock and key can be used in several different examples. All right. Function, environmental conditions. So here you have some crayons. You put them all together, and afterwards, the other day, I really excited about my new coloring book, so I brought a new pack of acrylic crayons. I forgot I left them in my pocket when I went to do my laundry later in the week, and what you see is what happened in this stack of crayons after they were put in a dryer. So they all melted together because it was too hot. So how would you describe what happens to crayons in image A? They melted together. Will I be able to accurately color my coloring book with crayons like this? No, it's still wax, by the way. So that should be thoughtful. They're melted, but they're not burned. So they lost their shape, but they haven't chemically reacted. So they're still colored wax. You can still use them to draw with, but they're not useful for drawing because the shape has changed, right? What caused my crayons to be changed? The heat, right? The shape changed because of the heat. What are some things that you've seen that melted or... Now, they use the word melted. In this case, that is what happened. It turned from solid to liquid and then went back to solid again. Have you seen melted change shape, right? Or change shape, so two different things, right? In which impacted their ability to work correctly. Come up with at least 3 examples of where the change in shape made it not work correctly again. I would say a plastic wheel of a car, of a toy car. If it melted and it turned into a square or some kind of other shape that wouldn't turn very easily, that would suck. Your toy car wouldn't work anymore. If you had a plastic plane, if you had a plane that, these horrible drawings. If you have a plane whose wings, let me redraw them down here. This plane wouldn't fly anyways, but if you had a plane that could somehow fly like that, but then because of heat, the plastic melted and reset, and what you ended up with was something like, that, then that wouldn't be any good. That wouldn't fly at all. So that's 2 examples. That's a 3rd example of something that would change shape and no longer work, huh? Okay. What about a car door? What about a car door? And the car got into an accident and because the car got in an accident, something else hit it and banged into it, another car banged into it, and because it banged into it, the door looked something more like, really smashed up. It wouldn't work anymore, right? If the door was all smashed up, it wouldn't work anymore. Not to mention the wheel's not round, but whatever. The point is, if the door no longer closed because it didn't fit, the shape was misshapen because of a car accident, then it wouldn't work anymore as a door, would it? So there's the 3 examples. I hope that helped. Above shows what happens when enzymes exposed to heat. How is it similar to what happened to my crayons? So you take an enzyme substrate, you expose it enough heat that the hydrogen bonds, which are much weaker than covalent bonds, the substrate doesn't change shape because the substrate is covalently bonded to itself. So that takes a lot more energy than the amount of heat required to break the hydrogen bonds that give the enzyme its secondary shape. When that enzyme is heated, it loses its shape, so the substrate doesn't bond to it anymore. It can't work. The enzyme can't work. Substrate's never broken up. So what happens? Enzyme was exposed to it loses its shape. It loses its shape. How is this similar to what happened with my crayons? It was the same thing, right? The crayons melted and changed shape. Here, the protein didn't really melt in the sense it didn't become a liquid, but the hydrogen bonds were broken and it did change its shape. What term is used to describe what happened when the shape of the enzyme has been altered? We use a term. I hope you know it already. It's denatured. Right. Predict how this alteration will affect the enzyme's ability to function properly. The enzyme won't work. Using figure A and B. So here's A, here's B. Here's optimum temperature of a human enzyme. This is the optimum temperature of an enzyme in a hot spring. Now, it's interesting that this enzyme, obviously this is about the body temperature of a human. Average body temperature human is the optimum temperature of the enzyme found in humans. That makes sense. This is the temperature at which enzymes in hot springs exist. And so what we find is that their enzyme, the bacteria in hot springs exist here at this temperature. So it makes sense that their enzymes work at this temperature. But then they stop working when it gets too hot, and ours stop working when it gets too hot. When it gets hotter than our normal temperature, than our average temperature, when it gets hotter than the bacterium's normal temperature, its warmest temperature. All right, what are 2 conditions that affect enzyme? What are 2 conditions that affect enzyme function? This was looking at temperature, but here you're looking at pH. And so hydrogen ion concentration and salt concentration. So any ion concentration can disrupt hydrogen bonds, therefore disrupt the shape of the protein. So here's the pH of the for pepsin. This is a pH normal for tripepsin, and what you find is really tripepsin is found in more normal conditions, where pepsin is found in more acidic areas. If you're looking at the human body, where might you find pepsin? What do you think? Where might you find it? Pepsin? Well, I hope you know that the human stomach is acidic, where most of the rest of the body is not acidic. So you're likely to find pepsin in the digestive system of humans, where you might find tripepsin if it's found in humans, right? You'd find it. You might think you would find it where? Maybe in the blood or some other organ that has a pH that's closer to 7, which is neutral, right? Not acidic and not basic, versus 2 or 3, which is the acidity of the human stomach. All right, what are the 2 conditions that affect enzymes that we see here? I hope you know it's temperature. The temperature is affecting the enzyme rate of reaction, and the pH is affecting the rate of reaction. What does that have to do with enzymes? Rate of reaction is how we measure an enzyme. Because what's an enzyme doing? Making the reaction go faster. So temperature and then pH. Provide an explanation for why human enzymes function best at 40, but enzymes for offsprings work at 70. As I said earlier, this is where these hot spring bacteria live, at 70 degrees. Why would you expect it to have enzymes that don't work at this temperature? That wouldn't make any sense. And humans, our average temperature is around 35, 37 degrees Celsius. So it makes sense that that's the temperature at which our enzymes work. It's reasonable. Okay. What do you think happens to a human enzyme when the temperature increases to 45? Well, it denatures, right? It loses shape. In 8b, you can see two different enzymes. Why do you think they work best at different pHs? Because this might work in a place where it's very acidic, and this will work in a place where it's not, that it's neutral. So this might work best in the stomach of a human, and this might work best in the blood of a human. When in actuality, what it is that pepsin is found in the stomach. It's released by cells in the stomach to help break down materials, where tripepsin is found in the small intestine, further down in the digestive system, where the acidity has been neutralized. So works at acidic pH, where this one's further down the intestinal tract, where it's not as acidic. Enzymes are complex proteins produced by all living things. Enzymes are substrates that help. Enzymes are substances that help carry out a number of the body's functions, like transforming food and chemical elements into other needed substances. Enzymes can help eliminate toxins in the colon, kidneys, liver, lungs, and skin. Your body has millions of enzymes controlling millions of tiny reactions. Under normal circumstances, each enzyme is able to catalyze many reactions before they stop working. Like all proteins, enzymes consist of chains of amino acids linked together. The amino acids within each kind of enzyme have a characteristic arrangement. The bond between the different amino acids in the chains are weak, may be broken by such conditions as high temperatures or pHs, or changes in pHs. When the bonds are broken, the enzymes become non-functional. Sometimes it results from disease. Sometimes this results in disease. So I'm not going to draw the whole thing out again, but we've done it numerous times. You have these chains of amino acids that are covalently bonded together. We call these bonds peptide bonds. I'm not going to draw out the amino acids again, but each of these have an R group. The R groups could be the same, or they could be different from one amino acid to the next in the chain. But there are 20 different options for your body to choose from, and each of them is different. And you might get hydrogen bonding, which is that weak interaction happening between two of them, which would then bend this molecule into a different shape, right? So then you would have a protein. This is very simplistic, obviously. I hope it's obvious to you. Because of hydrogen bonding, you might have this weak attraction between these two amino acids, pull them into a specific shape. That shape is what we've been talking about. This shape, this attraction can be impacted by temperature, or by pH, right? Or by salt concentration. So all these things can impact the shape of that protein because the hydrogen bonds can be impacted by that. The most well-known important enzymes are the digestive system enzymes. Amylase contains saliva, splits carbohydrates in order to be assimilated by the intestine. Protease, contained in the gastric juices, splits proteins so they can be digested. Lipase, secreted by a pancreas, splits the fats into fatty acids so that they can be digested. What are some uses for enzymes? Well, it seems fairly general. But for 21, I would say they can do just about anything. They can break things up, break up molecules that we call substrates. Or they can, that's A. B, they can build up molecules. By helping the parts of molecules get put together, also known as substrates. So they can build proteins or they can break up proteins. They can build nucleic acids or break them up. So in general, they can when they break them up in living systems, we usually call them, we say hydrolyze. When they're putting them together, we call them dehydration synthesis. Water goes in for hydrolyze. Out comes water when you're synthesizing something. But notice that you need an enzyme to do these things. That should make sense to you because you don't want your proteins to start falling apart as soon as they hit water, right? You don't want to see water melt you or change your, start to dissolve you, right? So you need these proteins to stick together. You don't want them to be hydrolyzed simply because they're in water. So how do you control that? You require enzymes to hydrolyze, to break up these polymers. Low-grade fevers are healthy because they help the body get rid of harmful bacteria and viruses. But when fevers reach a temperature of 105 Fahrenheit, it is extremely dangerous. Using the knowledge of enzymes to explain why. We've said this in class numerous times. Once you get up to 105, enzymes start to unravel. All your enzymes start to unravel because way beyond your normal range of temperature range, and so none of the enzymes are designed to work at this temperature. So they lose their shape, and if they lose their shape, they can't do their function. You can't hydrolyze molecules. You can't synthesize molecules. You can't make the proteins you need. You can't break down the proteins you need to in order to do digestion. There's absolutely all your bodily functions cease after a while at 105 degrees Fahrenheit. Do you feel you understand the role of enzymes in living things and how they work? What questions do you still have about enzymes? Well, there's a lot to learn, but I hope that you have at least the basics down. I hope that you understand that, one, enzymes make reactions go faster. All right. Two, oh, by the way, by lowering the activation energy. I hope that's clear. 2 enzymes are not used. Three, the substrate is what the enzyme works on, either to break it up or to build it up, whatever it's doing. The substrate's the thing it's working on. The substrate is used up by the reaction. The enzyme shape is important. Six, the enzyme must fit the substrate. Seven, temperature and pH / salt concentration does impact the enzyme shape. Therefore, the function. So how fast it goes, the two impact each other, right? So if you change the pH or the temperature outside that normal, it's going to impact the function. Well, I hope that helped.